And welcome everybody. I'm very happy to announce our first speaker for today, Jonas Ries. He studied physics in Germany at the universities of Constance and Bremen, and after this he went to the TU Dresden for his PhD in biophysics. In 2009 he changed as postdoc to the ETH Zurich, and in 2012 he became group leader at the EMBL in Heidelberg. Since 2023, he is now professor at the University of Vienna. Jonas Ries made many important contributions to the field of super-resolution microscopy. Lately, for example, by demonstrating MINFLUX tracking of kinesin in living cells. So I'm very much looking forward to his tutorial on single-molecule localization microscopy and MINFLUX. So, Jonas Ries, the stage is yours.
Thea, thank you so much for this really nice introduction. Also a big thank you to the organizers for inviting me here. Do you hear me in the back? Okay, if I tend to wander around, so if I ever lose the microphone, please signal me. So, when I prepared this tutorial, I tried to prepare it in a way that it's useful and understandable, even for those who don't have a background in super resolution microscopy, but also maybe interesting for those of you who are already using these techniques. So you all heard about the resolution revolution in structural biology, especially electron microscopy, electron tomography, that now allows us to solve structures of proteins even inside cells, and that with angstrom resolution, and that's absolutely amazing. However, to reach such a high resolution, we still have to typically average many different particles, and this is not so easy to do if you look at more dynamic and more heterogeneous protein assemblies. Also, we need such a high resolution to identify specific proteins in the electron densities. So this is why I think that optical super resolution microscopy can be the ideal complementary techniques to these structural biology techniques. Of course, the resolution is a bit lower. It's typically limited by the size of the fluorophore you put on your target protein to a nanometer or so. But we have the big advantage that we have the molecular specificity of the fluorescence label. We can detect this label fluorophore with this very high contrast. That allows us to look at individual structures without any averaging. And at least in principle, we can do this in the living cell and directly measure dynamics, something that the other structural biology techniques cannot really do. So in my group, we are developing now super resolution microscopy techniques with the aim to measure the structure and the dynamics of molecular machines ultimately in the living cell. So here you see an overview of the resolution improvements of electron microscopy and optical microscopy over the last century. You see that the resolution for electron microscopy really increased dramatically during the last century. But if you compare this with light microscopy, the resolution revolution is really happening here in the field of optical microscopy. Here in this graph, I denoted by crosses those techniques that we call single molecule localization microscopy or related techniques which are based on determine the precise position of single molecules. And this is what I will talk about today, kind of the techniques like Palm and Storm, DNA paint, but also MINFLUX. But before I tell you how these techniques work, let us take a step back and just talk about the resolution, the diffraction limited resolution of light microscopy. Because light is a wave, we cannot resolve structures much smaller than the wavelength of light because of this effect called diffraction that causes even a single fluorophore to look blurred in our microscope, either in the eyepiece or on the camera. The problem is if we have now two small objects that get close together so that these blurred spots overlap, at some point we cannot tell them apart. We cannot resolve them anymore. And this happens at around the wavelength over the two times the NA, the numerical aperture of the objective. So for 100 years and more, people thought this is the best we can do. It will never be possible to resolve structures with light better than given by this equation. It's set literally here in stone in the memorial for Ernst Abbe in Jena. But then in 2006, the so-called super resolution microscopy methods were developed. Of course, they cannot ignore that light is a wave, but they found smart tricks around this limitation to increase the resolution. So today I will mainly talk about the technique developed by Eric Betzig and others that we call single molecule localization microscopy. So this These techniques are based on the fact that it's much easier to localize single fluorophores or small objects than to tell them apart, than to resolve them. Because if we have a fluorophore far away from other fluorophores, we can determine the precise position by just looking where the center of this blurred out spot is. We typically do it by fitting a model of the Poinsett function to the data. So the precision with which we can resolve single molecules is described by this relationship here. So the localization precision scales with the old resolution limit divided by the square root of the number of detected photons. And these number of detected photons is really what matters most in single molecule microscopy. It is our currency, and it's always what limits the resolution. And the reason for this is, of course, shot noise. Because of the randomness of the photon detection, we don't always detect the same number of photons, but it varies over the pixels of our camera, and this leads to additional noise that limits localization precision. There's a very easy way to derive this equation, and for this, let's assume we have a microscope with a camera that can detect single photons and that we run very quickly, so we really see only single photons, and we have a pixel size that is smaller than the Poinsett function of our microscope. And we look at a single fluorophore with this Poinsett spread function. Then we detect photons at random positions, but within the Poinsett function. The Poinsett function itself is the probability density of detecting a photon. So we will detect more photons in the center of the Poinsett function. Now the standard deviation of the photons that we detect is then the same as the standard deviation of the Poinsett function, and we can calculate the average most likely position of the fluorophore by looking at the mean of the positions that we measure. Now we can ask the question, what's the error of this mean, of this estimation of the position? For this, we can just use our standard error of the mean, and we find that the localization precision, the error we do in the coordinate measurements, is the standard deviation of the Poinsett function divided by the number of measurements, the number of detected photons. That's precisely what we had. So this is very simple, but often the world is more complicated. There are more confounding factors we want to take into account. And then we can calculate more accurate estimations for the localization precision using fish information theory, the Cramer-Rao bound. I don't want to present you the equations here. It's something that you can look up and that is really useful to do. I just want to give you an idea how this works. So one way of determining the position by fitting of a Poinsett function is maximum likelihood estimation. This works by having a model for the Poinsett function and 3 parameters, for instance, the x and y position of the fluorophore, and then you calculate the likelihood that this model explains the data. And by varying the parameters, you can maximize this likelihood, and you end up with the most likely parameters, the most likely positions. Now, to understand what is the variance, the error we make in these position measurements, we can look at the likelihood at the maximum and see how strongly the likelihood varies when we change the parameter, the position, and then ask within the noise that we have in the measurement, the shot noise, for example, what range of parameters is still compatible with our measurements. And for example, if we do this for single molecule localization microscopy and take into account something like the limited size of the camera pixels or background, a fluorescence background, and this is derived by Mortensen here, we get this a bit more complicated but still understandable equation. We have a small modification on kind of the size of the Poinsett function, still 1 over square root of n. This is something you will see all the time, and then the background correction term. So if you now take typical photon numbers for fluorophores, around 5,000 photons, you see that we can localize fluorophores with a quite high precision of a few nanometers. That's great, but it does not really help us in biology, where we don't have much less than 1, but typically tens or hundreds or thousands of proteins within 1 diffraction limited spot. So we cannot really do this localization. And this is where the trick of Eric Betzig and others comes into play for single molecule localization microscopy. So assume we have a biological structure like this that looks blurred in our diffraction limited microscope. The trick we now play is to not look at all the fluorophores at the same time, but only to look at a small fraction of the fluorophores at a given time. And that we are able to do this, we need fluorophores that we can switch between a bright state and a dark state. A bright state that gives us as many photons as possible, and a dark state that is dark for a long time, so that we can have many individual single molecule detection events. So we detect the position of these fluorophores, switch them off, switch on another subset of fluorophores. This all happens completely randomly, stochastically. And in the end, the positions of the single fluorophores will represent a picture of this original structure. At this point, I really like to show this movie by Ricardo Enriquez. You all know this, I guess, which shows the Eiffel Tower, which you might know during the night blinks. The light bulbs there blink once an hour. And Ricardo took this movie, went home, and used the software he uses for single molecule localization microscopy to determine the position of every single light bulb. And on the right-hand side, you see how the positions of the light bulb fill in the image of the Eiffel Tower. Now, if you look very closely, you see that this edge here is really sharp. It's sharper than the images of the single light bulbs that you see. So we already have super resolution here, but of course, on spatial scales, not really usable for biology. But the same, of course, also works in the cell. Here you see on the left-hand side a movie that shows these single fluorophores. And on the right-hand side, you see how these single fluorophores slowly fill in the super resolution image. Now, we can detect hundreds, thousands, tens of thousands of camera frames. And at some point, we've seen most of the fluorophores, and we can then reconstruct a super resolution image with a resolution 10 times, 50 times, and nowadays even 100 times or more better than the diffraction limited resolution of our microscope. So to recapitulate this again, the quality of these super resolution images depends on one hand on the number of photons that we detect per fluorophore. It's the localization precision. But there's another factor, and that's the labeling density or the labeling efficiency. For continuous structures, in order to resolve them precisely, you want to sample them very densely, and then the Nyquist criterion applies, and we want to have a labeling density about 2 times higher than the desired resolution. Now, in biology, very often we reach spatial scales where this is just not possible because we have individual proteins that are arranged and we want to know where these proteins are and don't have continuous structures. And here I think it's more useful to speak about the labeling efficiency. What fraction of the target proteins do we see? And if this labeling efficiency is too low, we will end up with just a few dots, and these dots might tell us less than a diffraction limited image. So this technique, when it was developed independently by several groups and improved by others. This technique has been given many different acronyms, and the list nowadays probably goes on even longer. This is why I think it's quite useful to call them with a common term, and this is nowadays, I think, single molecule localization microscopy. Although some of these terms are still used to denote specific variants of this technique. For instance, PALM often is associated with photoconvertible proteins, DSTORM with organic dyes in a blinking buffer. I'll come back to this. So the development of this trick of using blinking fluorophores now allowed us to overcome the resolution of optical microscopy, the resolution limit, so we end up with a new super resolution limit. So that brings me to the outline for today. I will, during the first half, talk about single molecule localization microscopy, and during the second half about MINFLUX. And I will start by explaining some fundamentals and then give some examples for mostly tools that we developed in the lab. So here you see an overview of a typical workflow for single molecule localization microscopy. So we have to prepare the sample with the blinking fluorophores, then we do the imaging, we have to fit the positions of the single fluorophores, then typically we do post-processing to correct for residual drifts, to, for example, merge localizations that persist over several frames into a single localization, filter out bad localizations, and to assign a color. From this, we can then render a super resolution image, but we can also take the output of single molecule localization microscopy. These are the coordinates of the single fluorophores directly for further analysis. So by far, the most critical step in this protocol is the biology, is the sample preparation. I think in 90% of the cases where experiments don't work, it's because the sample is not suitable. But then you see that also a lot of the workflow is data analysis, from the fitting to the analysis. And for this, there are a few software packages out there. I think one of the most commonly used is an ImageJ plugin, Thunderstorm. Then there are several others out there. The Jungmann Lab has a nice software, Picasso, and also we have our own SMAP that includes already many routines for this analysis of the single molecule data. So now I will discuss the steps of this workflow step by step and give some of my ideas to this, and then present some tools to maybe improve single molecule localization microscopy. So let's start with the biology. So, as I mentioned, it's necessary for single molecule localization microscopy to use fluorophores that we can switch between the bright state and the dark state. And there are really many possibilities of doing this. One possibility is to use photoactivatable or photoconvertible fluorescent proteins. When the cell produces these proteins, they are dark or the wrong color, so we don't see them, but we can switch them on with a low dose of UV light, and then they become bright, we can image them, and then they bleach and switch off again. Another quite common approach is to use organic dyes, and typically something like LX647 is used in what is called a blinking buffer, where we do oxygen depletion and added thiol to induce long-lived dark states. But there are also other dyes that are really useful, for example, self-blinking dyes where you don't need UV light to switch them on and off, or photoactivatable dyes that have a photocleavable group that you can switch on once with UV light. And there's a third class. What is becoming more and more popular is use transient binding, and then it's called paint. And I think you all know DNA paint. And this works like this, that we label our target structure with a single-stranded DNA, maybe 8 or 10 base pairs long. And then we add complementary DNA to the buffer that carries a fluorophore. Now, when it's in buffer, these imaging strands are freely diffusing. They're completely blurred out on the camera. We don't see them. We might just see a slight background. But once they bind to the complementary DNA, they are immobilized and don't diffuse anymore, and we see them as a bright event. And so we have this on-off switching. A fluorophore binds, it's bright, unbinds, and then we just see the background of the freely diffusing imaging strands. So one advantage of using DNA paint is that we allow the fluorophores to give all their photons within this one on-switching event. We don't introduce this blinking, therefore these dyes can be very bright. Another big advantage is that we can easily do multicolor imaging sequentially. For this, we label the different proteins with different DNA strands, and then first at the first imaging strand, only complementary to the first protein label, image it, wash it out, then add the second imaging strand, let it bind, image it during the binding cycles. And after many cycles, we can we can even do something like 30-color super resolution. And this is some really amazing results by the Jungmann lab, where they imaged neurons in 3D in 30 colors and could beautifully resolve the relative arrangements of proteins here in the synapse. But how can we get our fluorophores to the target proteins, especially our organic dyes? So there are, again, many options, but I think the most common still is to use antibody labeling, immunolabeling, using a primary antibody and a secondary antibody. But you directly see what's the problem here. The fluorophore that might sit somewhere here is now very far away from the protein, and as an example, you see a microtubule. Therefore, now that we have this high resolution, we don't want to limit ourselves by the size of the label, so other smaller labeling schemes are preferential. We can use, as I said, these photoconvertible fluorescent proteins. We can use nanobodies that are really small antibodies, sometimes with a high affinity. And something that we like to use in my lab a lot is these enzymatic labeling schemes like SnapTag or HaloTag. They are fused also to the target protein of interest, like a GFP, but then we can add from outside dye, sometimes even membrane-permeable dye, that binds to the proteins, and this dye only has a very small molecule attached to it, so it can even penetrate densely packed protein structures and leads to better labeling efficiencies compared to antibodies. If you want to be even smaller, if you're lucky, you find a molecule that directly binds the protein of interest, like phalloidin for actin or paclitaxel for microtubules. Or you can do something like genetic code expansion and use unnatural amino acids to replace individual amino acids by a clickable version to which you can then attach a fluorophore. But this is still quite cumbersome to do, and it's difficult to do it stoichiometrically, to have it, if you do it endogenously, to have it at the endogenous level. Now, how about live cell single molecule localization microscopy? This is kind of how I sold this to you in the beginning. Super resolution microscopy is great because we can measure dynamics. Well, indeed, it's possible, and here you see nuclear pores that we imaged in the living cell, but it's still quite limited. So one of the main limitations is speed. Remember, we have to image these tens of thousands of camera frames to reconstruct one super resolution image, and this really needs time. Also, another problem is that nowadays the fluorophores that work well in the living cell often can be switched on and off only once. And that means we have to distribute the fluorophores among the camera frames. And if I do this for this example and just divide it into 5 frames, you see that each of these frames really only carries much less information than the whole. But arguably, the biggest problem is phototoxicity. I come back to this in a second. But because of all of these problems, many groups, including us, do mostly fixed cell single molecule localization microscopy. So cells develop to withstand something like sunlight, which is a kilowatt per square meter about, or in microscopic units, a nanowatt per square micrometer. And as some of you might have experienced, even sometimes you experience phototoxicity in sunlight. So the intensities that we use in single molecule imaging or in fluorescence microscopy in general are typically orders of magnitude higher. And this has been studied. For example, here, this study here looked at it's called plating efficiency, if cells still divide and duplicate properly, and then they quantified which laser irradiation causes 10% loss in plating efficiency, so in proliferation of the cells. And they found you can do maybe 10 seconds of single molecule imaging, not really enough for localization microscopy, take 10 confocal images, hardly a stack, and not even a stat stack. So this sounds bad, but on the other hand, there are many processes that are not directly affected by phototoxicity, or much less so. And if the process that we are looking at is not affected, we don't care if the cell still divides later, or even if it blows up 5 minutes later. But this is really a warning. It is very important if you do live cell super resolution microscopy to always test for this, to always make sure that phototoxicity doesn't affect what we are looking at. So how do you take this data? So what microscope do you need? Well, actually a very, very simple microscope. Basically, you need only an objective. You need a laser to excite the single fluorophores, because typically lamps or LEDs don't have the laser power we need to see single fluorophores easily. You need a dichroic, a filter, and your tube lens and a sensitive camera. And that's it. You can do single molecule localization in epi, illumination, turf, or high-low, this highly inclined light sheet to reduce background. All of this works very well. But I think something that is really essential is that the microscope is very stable. Imagine we now have a resolution of a nanometer or 2-3 nanometers and measurement times of an hour. And it's really not easy to build a microscope that is stable on the nanometer scale over an hour or longer. So this is why mostly people use active Z stabilization. I think this is really required, because otherwise Z drifts at some point make you lose the area of interest altogether, because you get out of focus. But also any Z drift can lead to XY drifts that are not homogeneous across the field of view, because the Poinsett functions might be slightly different. So I think Z stabilization is really required for good localization microscopy measurements, but XY drifts can then often be corrected in post-processing, so one doesn't need active stabilization there. It gets a bit more complicated if you want to do something like 3D or multicolor super resolution microscopy, but only slightly. For example, 3D single molecule localization microscopy. So when we localize our single fluorophores, we get from this localization the X and Y position, but no direct information about the Z position. And this is a problem. Biology is intrinsically 3-dimensional. Proteins are arranged in 3D. And that's why quite early on, single molecule localization microscopy was extended to 3D. The idea here is to not only look at the positions of the single fluorophores, but also the shape of the single molecule images, and to extract the Z position from this shape. And this is typically done after introducing aberrations in a defined way. We call this Poinsett function engineering. And the most common approach is to just insert a cylindrical lens. This lens then focuses the light more strongly in one direction, let's say in the X direction, then in the Y direction. And this leads to these elliptical Poinsett functions. And by just looking at the ratio of these ellipses, we can calculate the Z position. Here you see an example from my lab where we imaged microtubules using DNA paint. And you can nicely see kind of this in 3D, this hollow structure of the microtubules. But then if you look at the scale bar here, you see something is wrong. Microtubules we know have a diameter of maybe 25 nanometers. Here the size is 50 nanometers or more. So what happened here? So the reason is that we used antibodies for labeling, and they, as I showed you, introduced this linkage error. So we create this halo of fluorophores around the microtubules, and the microtubules themselves are here in the dark center. So if you want negative stain, super resolution microscopy. Now we can measure this high resolution in 3D in a slice, maybe 800 nanometers to 1 micrometer around the focal plane. And in this slice, we can get 3D resolution without scanning, without moving the sample or the objective. We just infer it from the shape of the single fluorophores. But if you want to go deeper, have a larger Z range, we need to image in super resolution many slices and then to stitch them together. And here you see an example where we did this, again on microtubules, to image the microtubule skeleton in an entire cell. So this was 3D, now multicolor. If we look only in a single color, we typically learn much less about the target structure than if we can do it in 2 colors simultaneously, because then we can directly relate different molecules to each other and their relative arrangement. Now there are many ways of doing multicolor super resolution microscopy. We can do it sequential, as in DNA paint, or by using 2 spectrally very different fluorophores that blink and can be activated at different wavelengths. But something that I think is very useful and that we like to do a lot in my lab is called ratiometric or demixing single molecule localization microscopy. The idea here is to label your target structure with 2 dyes that are spectrally very similar and that have similar blinking characteristics in the same blinking buffer. Then you image these 2 fluorophores with 2 spectral channels, and you split the spectral channel somewhere between the fluorophores. But for standard microscopy, that would be a horrendous mess. You couldn't use this at all because of the super strong crosstalk, because you see both fluorophores basically in both channels. But in single molecule localization microscopy, we have the advantage that we know we look at single fluorophores only, and they can be either one color or the other color. So we just have to look at the intensity ratio between these colors to uniquely assign the color, between these 2 channels to assign the color. The big advantage here is that image registration is trivial because we see both fluorophores in both channels. And this is in contrast to something like sequential imaging where drifts lead to then displacements that have to be corrected for. Or if you use completely different colors, we can easily have chromatic aberrations that exceed the 5 or 10 nanometer spatial scale. We can extend this demixing approach to 4 colors, still using only 2 spectral channels. Then we just have to look at the intensity ratio of the fluorophore in these 2 channels. And this is what we did here for 4 proteins in the nuclear pore complex. So down here you see one image acquired simultaneously in all 4 colors of a single nuclear pore, and up here an average that we calculated from, I think, 1,000 individual particles. So now I want to show you a few tools that we developed in the lab that might be useful. And the first is about quality control. So I think quality control is something really important if you want to know if our microscopes are still as good as they were a week ago, or if our microscope is as good as those that they say Nikon sells us. And here, for example, you see 2 publicly available data sets that show the same sample, microtubules, and image with the same technology, but they don't really look the same. And the reason for this can be manifold. It can be the microscope optics, the imaging conditions, the fluorophores or labeling approach that we used, the sample preparation protocol, what fixation did we use, or even the software that we used for analysis. And to investigate all of these points and to optimize them, what we really would like to have is a reference standard, a standard sample, a sample that is the same every time and everywhere we look at it. Therefore, we developed such a standard sample, and our idea was to use nuclear pore complexes. Because here nature puts proteins in a well-defined geometry, in a defined stoichiometry in the cell, and that in multiple copies. So we chose as a target protein the protein NUP96 that forms 2 rings in the nuclear pore. Each ring is occupied by 2 times 8 proteins, and we are lucky that the 2 rings are in register. That's why in the top view we can nicely resolve the 8-fold symmetry of the nuclear pore. Now we can ask questions, for example, can we resolve these 2 rings in Z? Then we know we have a Z resolution of at least 50 nanometers. So we then generated homozygous CRISPR cell lines with endogenous labels, with commonly used super resolution labels. For the physicists among you, that means we labeled the proteins in the genome, and we did it completely so that all the NUP96 proteins in the cell have a label attached to it, and we keep the concentration that the cell has with these proteins. And these cells now turned out to be very useful for us to optimize all steps of the data acquisition or even data analysis workflow. And they can also be used as counting reference standards to count the number of proteins in unknown complexes. And I would say they are now, or they have now become somehow the gold standard for quality control in super resolution microscopy. So now we can take images of nuclear pore complexes or many other cellular structures. They look beautiful, but then the question is, what do we do with this? How can we analyze these images in a quantitative and meaningful way to investigate our biological hypothesis? And when we were at this point, there was really not much out there to do. So we came up with our own approach, and this approach is to fit a geometric model directly to the raw data of single molecule localization microscopy, to the point clouds that we measure. For this, we have this model that depends on certain parameters that describe the geometry. Then we calculate the likelihood that such a model explains the data. And then we maximize this likelihood by using an optimization algorithm. And we end up with the most likely parameters of the model that explain the data. Now we can also use this to distinguish between different models and ask if this nuclear pore here is better described by an 8-fold or a 6-fold symmetric model. We just have to do this optimization and compare the likelihoods. Now I think this approach is quite useful because it allows us to extract meaningful and specific parameters from individual particles without any averaging. For example, for the nuclear pore complex, we could extract the radius of the ring, the distance between the rings, but also the twist angle between the two rings. When we did this for thousands of nuclear pores and then compared the experimental data to realistic simulations, we found that they were not really the same. Especially the radius, but also the twist angle had a much larger distribution in the experiment compared to the simulations. And this tells us that indeed there is heterogeneity. Not all nuclear pores are the same. It's something that has been known and is becoming more and more apparent, and something you also see by just looking at these images. But this is now a way to quantify this precisely. So we can also use this approach for single particle averaging, something that of course is very common in electron microscopy, single particle averaging, although what we do is more related to, let's say, subtomogram averaging. The idea is to always take two experimental particles and denote one the model, the other the data, and use our framework to look at the best overlap between these two, to look at the relative positions and rotations. If we do this now in a smart way for all the particles in our data set, we can align them on top of each other and generate these kind of probability distribution maps for the proteins. Now, if you look at the upper and lower rings separately, you see some nice features. You see that these corners are not point-like, they're a bit elongated, and they point outward in the spike-like fashion. You almost see here two individual dots, and this directly represents the fact that each corner of the nuclear pore complex is occupied by two proteins with a slightly different radius. I should say that we can calculate these averages now without any assumptions on the geometry or symmetry in this case. We can also use this framework to register many data sets on top of each other to calculate these multicolor protein distribution maps. For this example, we always imaged our nuclear pores with our reference protein NAP96 and one additional protein that we used labeled with antibodies. By then registering all of the NAP96 particles, we automatically registered all of the other colors in each data set, but also could combine all the data sets into one common coordinate system. And I think approaches like this might be useful in the future to denote or to find out the position of proteins that might be too small or too flexible to be visible in electron densities. But for this to work, of course, the particles have to be more or less the same. We know that the nuclear pores are not really the same, but still when we align them, we get a more blurred out version of the averages, which might still be informative because these are then protein distribution maps. And this is in contrast to electron microscopy, where any heterogeneity blurs out the resolution beyond these, 0.5 nanometers that you need to assign protein identities to the electron densities. However, it doesn't work very well if you look at a very dynamic system. So I will now show you a bit how we can extend this kind of averaging to dynamic system, and we call it dynamic reconstructions. And I will do this on the example of a protein machine that we are quite interested in the lab. That's the machinery that drives clathrin-mediated endocytosis. So endocytosis is the process by which cells take up molecules from the surface and the environment. It's kind of how cells eat. So during endocytosis, 50 different proteins in many different copies assemble at the plasma membrane and then pull in the membrane and pinch off a vesicle. So this process has been quite difficult to study because of this huge complexity, the fast dynamics in the second regime, and the small size way below the resolution limit of optical microscopy. And as a result, we don't know very well where all of these proteins are during this endocytic process. So this we now are trying to solve using single molecule localization microscopy. Sorry. The idea here is to take many images in dual colors and then to use features in these images to sort them according to time, according to the process in endocytosis or pseudotime. Also, we can use these features to align all of these particles in space to generate these kind of composite images. And here is a dynamic reconstruction, just a proof of concept that shows the plasma membrane. DAS17 is an actin nucleating factor, and then an actin binding protein in red. So this reconstruction now runs in real time, and it would be not so easy to take a similar super resolution live cell movie, even in one color. So we also extended this to mammalian endocytosis, where clethin forms these beautiful cages, as you see here. And by imaging the clethin together with a second protein, here dynamin, it's a protein that assembles at the neck of the endocytic process and then pinches off the vesicle. We can use clethin to align everything in time and space to generate these kind of dynamic reconstructions that show us how dynamin gets to this point of action. So I think in general, this kind of these kind of dynamic reconstructions can be useful because they give time information to all the super resolution techniques that are intrinsically not compatible with live cell experiments. This is something like our high resolution single molecule localization microscopy images, DNA paint, but even something like expansion microscopy. But of course, it's also quite dangerous to do these reconstructions because we have to have the assumption that we look at one and the same underlying process. And if this is not the case, if we have different processes, we easily get artifacts and can reconstruct nonsense. This is why I think it would be very useful to push this towards real dynamic super resolution imaging. So one limitation that we have for this is, of course, the slow speed. And I want to show you how we can maybe do a bit better here. So as I mentioned, single molecule localization microscopy is a very slow technique. It takes a long time to acquire all these frames to get all the localizations that we need. And this is not good if you want to do high throughput localization microscopy, looking at many different conditions, many different proteins, or if you want to look at live samples. Now there are approaches to improve this throughput. One is to automate the microscopes and then have to run them around the clock, and that's what we did for one project. And this works well. It gives us a huge amount of data, but it doesn't help us with live cell single molecule localization microscopy, where the process that we're looking at itself determines how fast we have to measure. Another approach that, of course, people tried, including us, is to use faster cameras, higher laser powers, to just acquire these camera frames in a much shorter time. We investigate this systematically by using our nuclear pore complexes, and unfortunately, we found that if we go very fast, that we not only use localization precision, we also lose localizations. And on the other hand, if we go slow, and this is about 10 times slower than we usually measure, we could further improve the quality and get a better localization precision. So in the study, instead of being able to go faster, we ended up going slower. So finally, we can try to improve the information content of each camera frame by switching on many more fluorophores. However, the problem then is that we start to get these overlapping point-state functions. There are fitting algorithms that try to take this into account, but typically they don't work very well because it's a very complicated problem, and they would only work well if we know the precise start parameters to do the gradient descent. Otherwise, we can easily get trapped in a local minima and don't really resolve the single fluorophores. So in a public challenge that benchmarked different software for fitting, even revealed that simple single emitter fitters outperform dedicated multi-emitter fitters for high densities. So and this is what happens when you have a single emitter fitter. So the fitter thinks it's only one fluorophore, but if two are close together, you get a localization somewhere in the middle. So to improve on this, we developed a new fitting approach using deep learning convolutional neural networks, and that we call DECODE. The idea is to use these camera images as an input for your CNN, and as an output use, and then predict as an output the coordinates of your fluorophores. So you probably know that to train neural networks, we need a huge amount of data where we have the experimental data together with the ground truth. Unfortunately, that's something we don't have in single molecule localization microscopy, because when we take these camera images, we don't know precisely where the single fluorophores are. But we are very lucky, and that is that the image formation process in single molecule localization microscopy is very simple and well understood. So we can easily simulate quite realistic camera images. For this, we position our fluorophores at random in 3D, and we do it at random that we don't train the network to learn any structures which then could lead to hallucinations. Then we apply an image formation model. We need the precise points width function, and then take into account the fluorophore blinking and some camera parameters like pixels and noise. And these simulated images we can then use for training. We feed them through the network, get the predictions, compare the predictions with the ground truth positions of the emitters, and then, as is done in deep learning, we use an optimizer to modify the weights of the network to reduce the loss. This worked very well, and here you see some results where we participated in this single molecule localization microscopy software challenge, where one can download simulated images. One does the fit-in, uploads the results, and the organizers of these challenges then compare the uploaded results with the ground truth and calculate performance metrics. So here, this is a complicated graph, but it's in the end not so difficult to understand. On the horizontal axis, we have the detection accuracy, basically what fraction of the fluorophores do we see, and on the vertical axis, we have the localization error. Now each dot is one algorithm, each color is a different approach, 3D using astigmatism, 3D using a double helix, and 2D imaging. And you see that decode really outperforms all other algorithms, often by a large margin, especially here on this high density data set. And this is quite useful because it now allows us to switch on fluorophores at about 10 times higher densities, and therefore to image 10 times as fast, and that without increasing the imaging laser power, so without increasing phototoxicity. So here we could image the endoplasmatic reticulum with a time resolution of about 7 seconds and look at changes that occur here. We now also extended decode to multiple colors, and that's unpublished still, to do, for example, 4-pi imaging, ratiometric dual color imaging, biplane imaging. So for this, we extended our network to now have 2 input images. It's also a bit more complicated, the network. And then we output, for example, the intensities in both channels to then assign the color. Now, it's not easy to register 2 channels on the microscope with single nanometer accuracy. Typically, the channels are related by a transformation that is not trivial, and that has to be calibrated, for example, using multicolor beads on a cover slip. So we do this calibration and inform the network about this transformation by telling it in each pixel how far the channels are apart from each other. And again, after training this network, which typically takes a few hours, we can apply it on our data sets, and inference then is very fast. It takes minutes. And also here now for the multi-channel challenge, this is the biplane challenge, DecodePlex outperformed other algorithms by a lot. Now we get almost 2 times as good in the localization error while detecting many more fluorophores. This allowed us here to do, I think, quite beautiful multicolor images of the endoplasmic reticulum in 3D and microtubules in 3D. On the right-hand side, you see the densities that we are dealing with now. Here's a dual color overlay. Yeah, you might say, you showed us much more pretty microtubules before. Why do you show us these crappy ones? The reason is not the imaging, the reason is the sample preparation. It took us about 3 months to carefully optimize the fixation to see both endoplasmic reticulum and microtubules at the same time. Because with most fixation approaches, you either see one or the other, which made us really understand how important proper fixation protocols are, and how easily you lose structures here. So we are quite happy with this result. And we can now move through side views and appreciate the 3D dual color resolution that we obtain here. Also, we performed dynamic imaging of microtubules, now with biplane 3D super resolution microscopy, again under quite high densities, and you see these dynamic changes of the microtubule skeleton occurring over the time of about 1 minute. I must admit this is more or less the maximum time we can look at it, because if you image for 2 or 3 minutes, we do see that light still induces phototoxicity and leads to changes of the dynamics and the organization of the microtubules. So now, whenever we do localization microscopy and want to fit, especially in 3D, we need to know the point-spread function. Usually, we calibrate point-spread function by positioning beads on a cover slip and then taking a bead stack. So this works, but it's not then so easy to use this calibrated bead stack or this experimental bead stack for fitting of single molecules. So we therefore derived an approach to calibrate point-spread function models that we call UIPSF, or Universal Inverse Modeling of Point-Spread Functions. So the idea here is to have a model for the point-spread function that depends on parameters. In the most simple way, it's just a stack of images, and each voxel has an intensity that then denotes the probability in the point-spread function. Then we have the experimental data, for example, our bead stacks of many beads, and we use a forward model to simulate this experimental data using our point-spread function model. And in this very simple example, the forward model is just a shift of the point-spread function to the position of the beads with subpixel interpolation. We then can compare simulated images and camera images. In the beginning, of course, they don't look anything alike, but we can calculate the difference to calculate the loss, and through an iterative process, we can minimize this loss. And after many iterations, then the parameters converge to the right parameters. So we get then realistic point-spread functions and realistic positions of the beads. We don't have to use it using voxel-related point-spread functions. We can also directly learn the pupil function, or even the pupil function expanded in the basis of Zernike polynomials. These are the aberration modes. The forward model is then the scalar or the vectorial diffraction theory to calculate how single emitters look like. And this is quite useful because compared to the voxel-related point-spread function, we now have to learn much fewer parameters, especially if you use Zernike polynomials. This allowed us to even learn point-spread functions directly from the raw data in localization microscopy, the single blinking fluorophores. So we don't need to take any bead calibrations anymore. This is useful because we avoid one step, but mostly because the bead stacks don't always reflect how the single molecules look at. There might be different aberrations. It's typically a different cover slip. And using this approach, the in situ calibration of point-spread functions allows us to calibrate the point-spread function where we use them for measuring. This is illustrated here, where our collaborator, Ximing Li, took pictures of the nuclear envelope, and this time of the upper nuclear envelope, which is about 5 micrometers away from the cover slip. Then they used oil objectives, and going 5 micrometers into a sample using oil induces quite a lot of spherical aberrations. Then if you use the point-spread function calibrated from bead stacks, you see that at some point it works a bit, but then all the localizations are kind of collapsed into one plane. We don't get any 3D resolution. So in contrast, when we use the in situ PSF, we avoid this kind of deformation, this squeezing. We do get much better data, but of course the spherical aberrations reduce the contrast in the point-spread function, so we do lose compared to having a well-indexed match medium. Still, I think this is quite useful whenever you expect aberrations. We implemented UIPSF using TensorFlow. That's the platform, or one of the platforms used often for deep learning. But we don't use deep learning here. We only use the TensorFlow framework to do the automatic differentiation for us. And it can do so analytically, which makes it very performant. For us, it has the advantage that it's very easy to write the code, to write the forward model. We just have to literally put in the forward model. Everything else is handled by TensorFlow. This allowed us to make the forward model also much more complex, to add drifts during the acquisition, intensity fluctuations, field-dependent aberrations. We could model a bead of a certain size to take this into account, this blurring by the bead, pixelation on the camera, and so on. Also, it allowed us to extend this modeling for different microscopy technologies, point-spread function engineering with astigmatism or double helix, a 4-pi microscope where one detects at 4 channels simultaneously, multi-channel localization microscopy where we can also learn the transformation, or lattice light sheet microscopy. So yeah, this is, I think, something, if you do high-quality 3D localization microscopy, is something you could try out. So With this, I'm at the end of the first part of this talk, where I talked about single molecule localization microscopy. And if you want, we could also take questions about this part now, before I move on to MINFLUX. Any questions? Yeah.
Thanks for your talk. So I'm curious, can you already fit the aberration directly in the 4-pi model, in the 4-pi modality with UIPSF already?
Yes, we can do this. So we can, yeah. It works especially well if you use beads and then do a phase scan in the beads. Or it's, yeah, might not be even possible, but we get the aberrations for pi. And we now implement it in a way that we try to use it even to align the 4-pi microscope, where we have deformable mirrors and then use the output of your IPSF to change the deformable mirrors to correct for these aberrations.
So still you need a bit this stack. You can't directly fit it with the single molecule data already?
We can. We do this, yes.
Oh, cool.
Yes. It works very well if you are not too high in density, if you have bright fluorescence, so it works better for, let's say, the D-storm approach compared to the Palm approach. But even for Palm, it works more or less correctly, especially if you use more or less reasonable starting parameters. Let's say you use a bead calibration from 2 months ago as a starting point and then do the optimization using single molecules. Any other questions? Yeah.
Thanks. I have a question regarding the decode. Can you use decode to like train a model for other like 3D like engineer points functions like double helix or tetrapod?
Yes, of course, and we did this in the paper. And also here, I think it is advantageous to use deep learning, because often when you have something like a double helix point functions where you have the 2 lobes, you easily get overlapping points at functions, because you have 2 lobes here, 2 lobes here. And with single emitter fitters, you have to go to ultra low densities to properly fit them. And with decode, it doesn't matter. As long as it's not super dense, you can have these overlapping points at function, and it still localizes and detects them properly.
Thank you.
Okay, then I go on, and there is time for questions afterwards. Or is there more? Any more questions? I don't see any. Okay. Now, let's go to MINFLUX. So, after the development of single molecule localization microscopy or super resolution microscopy in general, this story repeated. Everybody thought that's the best we can localize single molecules. It will never be possible to localize single molecules better than given by this equation here. But then Stefan Hell, who already had received the Nobel Prize for stat microscopy, had a very smart idea to overcome this super resolution limit. So before I tell you precisely how MINFLUX works, let's look at a very simple one-dimensional example and a task where we want to determine the position of single fluorophore by probing it with one beam of light, a pattern beam of light, for instance, a focused laser beam as in a confocal microscope. We park the laser beam somewhere in the vicinity of the fluorophore and now want to know where the fluorophore is. How can we do this? Well, we detect a certain signal n, the number of photons, that's the Poinsett function at the position of the fluorophore times, let's say, the brightness of the fluorophore. We detect this intensity, go to the Poinsett function, go down, and know where the fluorophore is, x0. Mathematically, it's just x0 is the inverse of the Poinsett function, number of photons, divided by intensity. For this to work, of course, we have to know the brightness of the fluorophore, I0. Now we can ask the question, what error do we make with this kind of measurement? For this, we just have to look at the error of n, and do error propagation, and we find that n is I0, the derivative of the Poinsett function at the position of the fluorophore, and then the error of the fluorophore. This is something you can easily understand geometrically. You see here the error in x and the error in the photons are related by the slope of the Poinsett function, the f to the x. Now we know the error of the photons, because these are single photons that we detect. This is the shot noise, and whenever we have this random detection of photons, they follow a Poisson distribution. And that means that the variance equals the mean, or in this case, the standard deviation equals the square root of the mean of the photons. If we plug this into this equation here, then we can solve it for delta x, and we get this beautiful equation that the precision with which we can localize a single fluorophore scales like 1 over square root of n, as always. And then we have a term that only depends on the Poinsett function shape, not anymore on I0 or anything else. And it's the value of the Poinsett function at the position of the fluorophore divided by the gradient of the Poinsett function at the position of the fluorophore. In general, we don't know the brightness I0 of the fluorophore, and we have to perform at least 2 measurements to solve for the 2 unknowns, X position and intensity. Now we can investigate what is better, using a maximum for detection or using a minimum for detection. So if you use a minimum, we can still have a high gradient at the position of the fluorophore, so a strong F, the derivative of F, but the photon numbers are much lower and F is lower. So it seems that localization with a minimum is indeed preferential. And indeed, when we take this very simple equation for a maximum like a Gaussian profile or a minimum like a donut, you see that the theoretical localization precision is really much better for localizing with a minimum. And this is precisely the idea behind MINFLUX. In MINFLUX, we don't use a camera for imaging our single fluorophores, but we image the fluorophores by scanning a pattern beam around it. But the same as in single molecule localization microscopy is a technique to localize and to image single isolated fluorophores far away from all other fluorophores. So it works very well when we have these photo-switchable fluorophores, like for localization microscopy imaging, or for single particle tracking at ultra-low densities. So in MINFLUX, the donut-shaped beam typically is scanned around the single fluorophore. We know the position of the beam. We measure the intensity of the single fluorophores, and from this we can precisely triangulate the position. We measure the intensity, and from this we get the position of the single fluorophores. So what's the error we make here? So in MINFLUX, the localization error is given by this equation here. L is the size of the scan pattern, and down here you have, again, square root of n. Compared to camera-based imaging or localization, we have 2 advantages. First, we have the 2's in the denominator that makes sigma smaller, but then most importantly, L, the size of the scan pattern, we can choose to be smaller than the size of the Poinsett function. And by this we get the high resolution. Now a question for you. What size of L would you choose to have the best possible localization precision? The smallest sigma. Anyone? Well, it's clearly L equals 0. No scanning at all. It would be nice if this was true. It would make it much easier to build MINFLUX microscopes, but of course this is not the case. On one hand, we neglected here background. That is always the limiting factor of single molecule detection and of MINFLUX. But also this equation is only valid if our target fluorophore is within the scan pattern. But at the beginning of the experiment, we don't know where the fluorophore is. So we have to start with a large L. So this problem was solved with an approach called iterative MINFLUX, where one first scans with a large L, determines the approximate position of the single fluorophore, then recenters the beam pattern, scans with a larger L, and after a few iterations, one has zoomed into the single fluorophore. And this dynamic feedback, this dynamic change of the position of the pattern and change of the size of the pattern is really, I think, essential to get the best resolution in MINFLUX. So with this approach, iterative MINFLUX, at least in theory, it's even possible to overcome this 1 over square root of n dependence, because now the localization precision can scan with 1 over square root of n to the k being the number of iterations. So of all techniques that we know, MINFLUX is the technique that gives us the highest localization precision for a defined number of detected photons. And this is, again, important to realize. MINFLUX is good for photon efficiency. So if you don't care about, if you have more than enough photons, you won't gain much by doing MINFLUX. And there are now 2 main applications for MINFLUX. One is imaging, and you see here a single nuclear pore complex that we could image with a commercial MINFLUX instrument, and you can appreciate the high 3D resolution that we obtained here. And the other is single particle tracking. And I will briefly explain both of these approaches. So here you have a comparison of published data using different super resolution techniques, MINFLUX, but also single molecule localization microscopy techniques. And they were all obtained on the same sample, the NAP96 reference standards. If you compare them, especially the Z resolution, you see that MINFLUX really does very well, but it's It does not do better than, let's say, 4-pi super resolution microscopy or this new approach, RASI, which is an extension of DNA paint that clusters together many localizations in DNA paint. But there's one big limitation in MINFLUX, and that is that it is a really slow technique. Already single molecule localization microscopy is very slow because we have to take all the camera frames and localize all these fluorophores. But at least we can do this simultaneously for many fluorophores on the entire camera chip. MINFLUX has to go step by step and localize every single fluorophore after the other. That means for large fields of view, it's rather slow. For example, to take a 3D image of just, let's say, 10, 20 nuclear pore complexes easily takes an hour or two, whereas with localization microscopy, within 10, 20 minutes, you can have an entire nuclear envelope with, let's say, 1,000 nuclear pore complexes. So it's not clear if there are many biological applications where MINFLUX imaging is really more suitable than in any of the other super resolution techniques at the moment. It really depends on your question and what you want to learn. One of these applications might become live super resolution microscopy, and I want to explain this a bit now. So already a while back, we took our nuclear pore cell lines to the group of Stefan Hell, and they imaged these nuclear pores in the living cell using this rather dim photoconvertible protein and maple. And indeed, the resolution, the precision we got here was much better than we could achieve in live localization microscopy. And the reason is, of course, that these fluorescent proteins are rather dim, and with MINFLUX, we get the higher localization precision. Now the question is, will we be able to use MINFLUX really for dynamic imaging to look at conformational changes or rearrangements of these proteins, let's say opening, closing of the nuclear pore complex, or in endocytosis, rearrangement of the clathrin lattice with the nanometer 3D resolution that MINFLUX offers, and that with a sufficient time resolution? To investigate this, let's recompare the speeds of localization microscopy in MINFLUX. In localization microscopy, to avoid overlapping point state functions, if you don't want to use decode, we have to activate the fluorophores at a regime where all of the pixels are dark most of the time. Only once in a while we can have fluorophores switching on, and we have these single molecule images. And this, as I explained, takes time. In MINFLUX, the situation at first glance seems much worse. We cannot do we have to go point by point. And also in MINFLUX, we need much lower densities in single molecule localization microscopy. Single molecule localization microscopy fluorophore a bit close by might still not hurt so much. We have multi-emitter approaches. But in MINFLUX, we have our target molecule in the dark center of the donut. So it's very, very dim, and we still get the information. And as soon as another fluorophore comes close by and just enters, let's say, the rim of the donut, it can already outshine our target fluorophore. So we need to work it really at ultra-low densities in MINFLUX. But if we then focus on a very small region of interest, MINFLUX can be very fast. For instance, a region of interest the size of the nuclear pore complex. In theory, we could even beat here the limitations of localization microscopy, that all of the pixels have to be dark most of the time, by a targeted activation and deactivation approach. So the idea would be to start with quite high UV laser power and an imaging laser to switch on very quickly a fluorophore, and as soon as it's switched on, we detect the on-switching event and directly switch off the UV laser to avoid switching on a second fluorophore. Then we can perform our MINFLUX localization, and at some point when we collected enough photons for a given localization precision, we could switch off the fluorophore with a high intensity laser pulse and are ready to repeat the cycle again, and therefore to really activate image one fluorophore after the other, that may be every 10 milliseconds or even faster. With this, it should be possible to perform time-lapse microscopy using MINFLUX with nanometer 3D resolution and with a fast speed. At the moment, I don't think it works yet, at least not with us. And the reason is, on one hand, that the microscopes might not be fast enough, but mostly that we lack labels that are light cell compatible, that can be switched on and off many times with light, that we can switch very fast, and that give us enough photons to do MINFLUX with. Good. Now I want to briefly talk about some artifacts and challenges in MINFLUX. So the measurement MINFLUX is really complex compared to taking camera images. We have to find candidates for single fluorophores using some kind of scouting approach. Then we have to scan the scan patterns around the single fluorophore, determine on the fly within a microsecond the position of the single fluorophore to recenter the the scan path and then go through a few iterations. And it's really not clear how experimental artifacts or imperfections cause, or how imperfections cause artifacts under this complex imaging modality. Another problem is that the MINFLUX microscopes are really complex to build and to use. This is just the excitation path of a really beautiful MINFLUX instrument by the Francisco Balzarotti group that can be perform 3D and dual color MINFLUX. But as you see, there are a lot of components. Here, very importantly, we have 2 electro-optical deflectors for fast scanning. But in addition to these fast scanners, every MINFLUX microscope also needs a slow scanner to follow single molecules, because these fast scanners, we perform detection through a pinhole, and you cannot descan with these fast detectors because of efficiency and also because of autofluorescence caused in these detectors. So the range is always very limited with these fast scanners. And as large-scale scanners, one then can use piezo's or galvos, which is not shown here. So we have these complex instruments, and that's why I think we are very lucky that the Iberia company produced a commercial version of these instruments. They're not very cheap, but they produce quite decent data, and also my group now has one of those instruments. So let's look at how some of these artifacts are generated in MINFLUX. First, let's look at the background. To do this, we can use the standard formalism, Fisher information theory, and calculate the Kramer-Rao bound, taking into account background. And this is what we did here. So the background in MINFLUX can come from 2 different sources. First, we could have an imperfect zero because of some misalignment, and then a fluorophore even at the center of the donut will emit photons. In this case, we find that the localization precision here scales with the scan size. At the beginning, it gets smaller, but then if we have background for very small scan sizes, it diverges and gets very bad. So there will be an optimal size for L. But a background can also come from autofluorescence, from nearby dyes and so on. And the difference is that this background doesn't come from the photon budget from our dye. What it does, however, is that it increases here the intensity without increasing the slope of the point spread function, and therefore we have kind of this limited resolution even for very small L. Now this was easy, but in general artifacts can be much more complex, and to test these artifacts we have to do simulations, and this was something which we just finished and put out on bioRxiv a few days ago, so you can have a look for our MINFLUX simulator. This MINFLUX simulator simulates more or less an Iberia MINFLUX microscope or any other MINFLUX microscope in detail, so with the scouting, the scanning, the position estimation, and the repositioning of the detection volume. We can define simulations either programmatic or by directly loading this Iberia sequence files to directly simulate an experiment. We have realistic models for point spread functions, either using the vectorial model or using experimental point spread functions. We simulate pinholes, aberrations, imperfect zero, or the bead size, and calculate CRBs. And we have a realistic model for fluorophores, including switching, flickering, bleaching, and movement like diffusion or stepping. And we can simulate many fluorophores together to simulate something like images. And we implemented several estimators to test which estimators are best for our imaging. As a result, we then get kind of the output, the estimated positions of the fluorophores, but also additional parameters, in this case for the stepping experiment, the position of the galvo and the EOD. We can get images out if you do MINFLUX imaging, or statistics on the performance. Now I want to briefly show you a few examples what we learned using the simulations. And the first thing what we looked at is flickering of fluorophores. So when we scan the MINFLUX pattern, we have to assume that the fluorophore has a constant intensity at all patterns that we scan, because we kind of look at the relative intensities for the triangulation. But this is not true in general, because fluorophores can flicker. They can have short and long-lived dark states on many time scales. This is especially true, for example, for quantum dots. Now here we simulated a fluorophore that switches between a dark state and a bright state every 100 microseconds. And we have localization times of a millisecond. Without blinking, our MINFLUX experiments would result in a quite well-defined position with a small noise determined by the number of photons we detect. But as soon as we take this flickering into account, the errors get much worse. Indeed, they get 10 times worse than without flickering in the simulation. One way to combat this artifact is to not scan the pattern only once, but to scan it many, many times, because then we average over this flickering, and this is what we simulated here. But we see that we really have to simulate many repeats in order to get close to the best case, like 20 repeats or more. And this is something that is not easy to do, at least with the commercial MINFLUX instrument, because it's just not fast enough for this. So next we simulated MINFLUX imaging, and it is now becoming more and more used to use DNA paint for MINFLUX. The reason is, again, the ease of doing sequential multicolor imaging, but also the large number of photons that we detect in each event. However, the imaging strands are still bright. They diffuse around, and they diffuse around on a time scale and spatial scale as our pattern scan. So within microseconds, the intensity can change because an imaging strand diffuses by. And indeed, our simulations of this mode where we have blinking fluorophores and this diffusing imaging strands show that we get a lot of invalid localizations all around, and also those that are valid, that they show a reduced localization precision. If we simulate the same again, but using invisible imaging strands, then the resolution, of course, gets much, much better. And this might be a good idea to, at some point, try this fluorogenic DNA paint that has been developed by the Beversdorf group with MINFLUX. However, it could be that the quencher that leads to these dark imaging strands cause additional flickering, which again leads to a problem, as I showed you before. So this needs to be tested. We also simulated MINFLUX with photoconvertible fluorescent proteins, and we found that, unfortunately, most of the fluorescent proteins are kind of lost during the scouting or before we zoomed into them because of the limited photon budget. And there we have the disadvantage that we can switch them on only once, and then they bleach and are gone. And looking at this is kind of what we get in the best case on average when we use photoconvertible proteins in our MINFLUX. One really has to then look at many nuclear pores to find a beautiful one, as I showed you before. If you use blinking dyes in a blinking buffer, the D-storm approach, it looks quite nice. So this is something that we usually use for our MINFLUX imaging instead of photoconvertible proteins. So again, it shows that it's not really clear if MINFLUX imaging is the best technique. If you want to do super resolution imaging, maybe DNA paint or standard storm might be a better choice. But there's one approach where MINFLUX clearly outperforms camera-based techniques, and that's single fluorophore tracking. So in single fluorophore tracking, one determines the position of the single fluorophore with the donut. Then in every iteration, we move the scan pattern to the estimated position of the fluorophore. By doing so, we follow the fluorophore through the cell with our donut. Compared to camera-based tracking, we have three advantages. First, the high localization precision of MINFLUX. Second, we get this high precision with few photons, so we can be quite fast. And third, we need only very few photons for every localization. Or in other words, the fluorophore spends a lot of time close to the dark center of the donut, so it's not easily bleached. So we get very long tracks. So we then used MINFLUX to look at the motion of a motor protein called kinesin 1, that, as you see here in this beautiful animation, walks on microtubules and drags cargo through the cell. It does so by this kind of hand-over-hand motion, where the motor domain performs a 16 nanometer step that leads to an 8 nanometer step of the cargo binding domain. So this motion really has been studied quite extensively using all kinds of single molecule techniques, but all of this was performed in vitro using purified proteins because we lacked the spatial and temporal resolution to observe this motion in the living cell. This is what we wanted to change, but before we turned to living cells, we looked at fixed cells using a so-called motor paint approach, where we fix the cells and add labeled kinesins from the outside that bind to the microtubules and start walking. The advantage here is that we can reduce the ATP concentration and slow down the motion. And what you see here in this movie here, especially if you look at the running mean, is the position of the cargo binding domain. You nicely see these 8 nanometer steps. Here, sidestepping event, 8 nanometer steps. Now we can ask what resolution did we have in this measurement, and for this we can look at the standard deviation of the position in a single step. And we found, if you look at the raw data, a precision of around 3 nanometers. But since we can now acquire hundreds to thousands of localizations in each step, the position of the step is determined by a much higher precision with a standard error of a mean of probably better than 1 angstrom for most of the steps. However, this is not really the resolution that we get here, because the microscope is not stable on the sub-nanometer scale, and also the microtubules themselves can still move on the scale. But still, I think this could be a quite useful approach in the future to map out, on one hand, conformational changes of proteins in fixed cells, very similar to what is done in vitro, also using MINFLUX, but also to monitor and map out microtubules in fixed cells with protofilament resolution. Next, we turned to living cells, and here we expressed kinesin with a halo tag and labeled it with a beautiful Genelia dye. When we focused onto a really small part of the cell and started our MINFLUX tracking, we obtained many tracks, like these ones is one of the tracks that we rendered as a super resolution image. You nicely see this localization clustering like pearls on a string with a 16 nanometer distance. This is, of course, the 16 nanometer steps that you can also appreciate here. The localization precision here was around 2 nanometers with a time resolution of about a millisecond. This is quite good compared to what is possible for tracking fluorophores with cameras. We can now look at the position versus time and identify the individual steps. For each step, determine the step size and also the dwell time. And when we did this for hundreds of tracks and thousands of steps, we found we could kind of for the first time characterize the precise distribution of these parameters in the living cell. So I think this shows that MINFLUX could be useful to study conformational changes of proteins in living cells. However, there's a strong limitation, and that is that we cannot distinguish external motion from internal conformational changes. So this works quite well for something with very regular movement, as we showed for the kinesin, where we know the stepping happens along a line. Or it's something completely random, let's say free diffusion, where we don't care about internal motions. But for everything else, for all other conformational changes, we would need an internal reference. And this can be solved by extending MINFLUX tracking to dual colors, to 2 colors at the same time. Because then we could, let's say, put a red and a green fluorophore at 2 parts of our protein and then look at the relative distance, and that hopefully down to nanometer precision in 3D and 100 microsecond temporal resolution. And this would allow us to fulfill one of the dreams of structural biology, to directly monitor conformational changes of proteins in the living cell. So we are working towards this, and other groups already have achieved this. We did this in collaboration with Iberia, who built us a MINFLUX microscope that can simultaneously perform two color tracking independently, so it's kind of like 2 MINFLUXes in one box. With this, we could monitor our kinesins, where we labeled both motor domains with 2 different dyes. And as you see here, sometimes the red dot is in front, sometimes the blue dot is in front. So we do see kind of this hand-over-hand walking, but the quality, as you see, is not quite there yet compared to what we see in the single color. But we're working on this. And I think we're not so far away from being able to really watch conformational changes of proteins in living cells. So with this, I'm done. Actually, I'm done very early, much earlier than I expected. So we have a lot of time for questions, but before I want to acknowledge all the people who contributed to whatever I showed you, most and foremost my wonderful group, before at the EMBL, now at the University of Vienna, and all the alumni, our great collaborators and funding that we have, and thank you for your attention. So, questions?
Sorry, thank you for that, Michael. Can you do dual-dye labeling combined with something like DNA plant and check them for dynamics of other things in this?
Combined with what?
Combined with something like DNA paint?
DNA paint.
And just use MINFLUX to track things like that, so you can track other things with DNA plant.
The problem is if you have blinking probes like DNA paint or storm probes, then it's very unlikely that both of the two labels on our target proteins are on at the same time. So either one or the other is on, and so we could not really look at the dynamics.
No,
right?
don't blink in them. Don't
Oh,
blink
just
them.
to bind them
Yeah.
and have them bound there. Sure, I think this is one way. It's not easy, I think, to do this inside the living cell because the imaging strands, they don't, or also the labels don't really go into the cell. So one probably can do this on the cell surface. And I think live DNA paint has been done on the cell surface proteins, and then there's no reason why not to track them. That's a good point.
Sorry, maybe I have a question for the more previous slides. You showed us the 3D image with astigmatism, also with biplane. I'm curious, how's the performance of biplane versus astigmatism case?
So I think there has been studies on this which showed that biplane might be a bit better. We in my group use mostly astigmatism for 3D because it's simpler. We only need to use one channel, and then if you want to do dual channels, we can do dual color easily. So we can use the two channels then for dual color. So it's simpler, and we don't see much of a performance drop. It is true that in astigmatic images, you have these deformed pointed functions. So depending on where you are in Z, you have a worse error in X or in Y. So you get sometimes these stripes. You don't get them in biplane imaging, where you always have these symmetric pointed functions. And depending on where you are, sometimes one or the other is more in focus. So you might have a larger range with a good performance. But in the end, astigmatism is something like biplane, but the two planes are just in the two different axes instead of two different halves of the chip. So it's very similar.
Yeah, then there you have multiple channel detection, right? Then your registration error probably also will play a role here.
It is, but we calibrate the transformation very precisely, either with beads, and of course there we can make an error, or with your IPSF, which learns the transformation during the optimization process. So this, and also since for biplane, but also for multicolor imaging, we really see the fluophores on both channels. There is not really a problem with registration. For example, for the multicolor imaging, we now chose a dichroic that splits quite in the blue, so it transmits quite well Alexa 647, but not so much CF660C, also one of the dyes. And most of the photons are detected in the long channel. So if you want to be really sure to avoid any kind of aberrations, we take the position information only from the long channel and use the short channel to assign the color. So this is something one can do.
I'm so glad to know that.
Good.
Thank you for the great talk. We just set up our first SMLM machine in our lab, and we are using the commercial modality from size. So I would have a question about post-processing. We have the size software, and as you said, they are Picasso or SMAT. So which software will you recommend us to use as first?
So, since I wrote the software, I would of course recommend my software SMAP, but I think the other one is probably as good. I don't really know what all you can do with Picasso. I think drift correction it can do, and then it depends on what else you want to analyze to look at what you want to have. Also, it depends if you want to fit the raw data again, or if you want to use the fit from the Zeiss microscope and then just analyze the data you have. If you use our software, I recommend you to spend a bit of time going through the tutorials and getting to know it. It's quite complex, so it's nothing one can do within a minute. But then I think it's worth going through this and learning how to use it. And if there are any questions or bugs, please write to me.
Thank you.
Oh, okay, thank you. So I have some questions quite related to us from the user side. So previously I tried different software for the post-analysis. Like Imaris is a kind of commercial software, and they also try a lot of different machine learning approach. For detail, I don't know, but it looks quite amazing. Like they kind of customize for different company, And you can also just input what the lenses you use and what the numerical aperture somehow. But yeah, for the detail, I don't know, but they have done quite good Z resolution. And I also see you have done something quite good results. So I just wonder, like, have you have some kind of benchmark compare your method with these, and how many parameters you actually need for using your software as an input?
So I have not used the Maris system. I think it's expensive, so I bet it better be good. And in the end, fitting is, yeah, everybody uses the same. Either one uses Gaussian models that don't quite get the resolution and precision that you will get with the experimental point-state functions. Something else, if you have a model mismatch, for instance, a Gaussian function or the wrongly calibrated point-state function, usually you still get an okay precision, but you get an error, especially in Z. So you get the squeezing, as I showed you with the nuclear pores and the IPSF. So it's something to watch out for. If the same algorithm is used, that's a maximum likelihood estimation with an experimental point-state function, then they should all kind of perform the same. And then it depends more on how you filter the data, how you render the data, how you do the drift correction, and so on.
Yeah, so one thing also related is like if I use your software, I know like for such images, it's quite related to your sample or what kind of the dye properties. So do you have these parameters like kind of freely for the user to input, like, for example, I can do some calibration for myself, like put my dye in the media and see the properties, and I do self-calibration for my later image for the sample, for example.
Yeah, this is, I think, the same for all software, that you need to calibrate your point spread function. And this you can do with beads. It works all right. Only if you really want to be very precise or go deep, have aberrations go deep into the sample, you need to do this in situ with your IPSF. I don't know if Imaris does the same. I could imagine that they can estimate point spread functions. Then there are not so many parameters that you can screw up. It's really, it depends all on your sample, but then kind of the only parameter that you can adjust a bit is the threshold, what the software thinks is a proper localization. And we usually put this rather low, that we get many localizations, even wrong localizations, and then filter later based on the likelihood, based on the localization precision. So to get then to only retain the localizations that are good enough for us. If you use something like decode, of course you need to know more parameters. You need to know the approximate density of localizations, the approximate background, the approximate brightness. And for this, typically we do a SMAP fit, a standard single molecule fit before, to estimate the range of these parameters. But it's not very crucial. So the only thing that really matters is the precise point spread function model.
Final question, sorry. So do you have some numbers, like for example, the limitation of the depth, like we should achieve, or like, I mean, because for the decode or something different or everything, we also worry about if we overly iteration or overly calculation the results.
Yeah, sure. Yeah. Decode also doesn't solve the problems with the model mismatch. So we unfortunately see that even if you are, let's say, 500 nanometers away from the cover slip, you already get deformations in that using the all objective. We also have the problem with TOF objectives that we collect critical angle fluorescence. When we do bead calibration, that's always there. If you go 500 nanometers away, we don't. So also there we have a model mismatch. So you get this kind of local deformation in Z quite easily. And whenever you do bead calibrations, sometimes you don't really care about this. Sometimes you can kind of calibrate this out by looking at 1,000 pores and plotting the distance versus Z, and then using this as a correction factor. But otherwise, it's quite crucial. What we like to do nowadays is, when we want to go deeper, is to use not a normal oil objective, but a silicon oil objective, and do index matching. So to try to put the refractive index inside the sample, the same as the immersion medium, and with this we avoid spherical aberrations.
Okay, thank you.
Thank you for the nice talk. I'm sorry if I missed that, but if we already know, you showed the fluorophore flickering makes the precision very worse in MINFLUX. But if we already know the property of flickering, like 1 millisecond or what percentage, like can't we improve the MINFLUX like precision as you showed?
Yeah, I think Well, you can try to reduce the flickering. If you don't work in living cells, often the buffer can be changed, or you use a different fluorophore that shows less flickering. And honestly, we didn't look at this experimentally. I think that's something the field has to do to benchmark flickering on these timescales. The other way, of course, you can iterate average over many more fluorophores. So if you don't have to be super fast and need a time resolution of, let's say, 10 milliseconds, and even with the Iberia instrument, you can average over 20 cycles to then try to average out over this flickering.
But, like the timing or the exposure time or some scanning time was already optimized for like, some conditions, or like how did you determine that? Like, is the fastest one, or?
So in our simulations, actually I can even show this. In our simulations, we use the standard sequences that are provided by the company, and they're already quite optimized. But we found, at least in silico, they might not be completely optimal. For example, when we looked at this is where we simulated diffusion, and we wanted to know what's the fastest diffusion coefficient that we can measure, because it's known that it's difficult to follow free diffusion with MINFLUX, and even membrane diffusion of small proteins is challenging. So we then did the simulation for different size of scan patterns and always calculated what fraction of fluorophores could we track for at least 100 localizations. Indeed, we found an optimum at around 100 to 150 nanometers. So going very small is not good if you have fast diffusion. And that we could then do for many different parameters, change these parameters here, what's the fraction, what we can still image, let's say the pattern size, the photon limit, let's say the precision you want to have, or the pattern dwell time. And with this, we could kind of get higher with the maximum diffusion coefficient. We now have to test it in the experiment if this is really true. But maybe, since I'm here, one point of caution. We found that if we are close to the limit that we can detect with MINFLUX, we have a strong bias towards slow diffusion. And also, the mean square displacement that we measure with MINFLUX is not linear anymore. So this could, if you use this, for instance, to study membrane diffusion, could mimic some kind of subdiffusion or so. Even if it's not there, just because of the limited time resolution and the tendency of MINFLUX to somehow smooth out the positions.
Yeah, so faster scanning is very, like it sounds like. Yeah. Okay, thank you very much.
So you mentioned the the commercial MINFLUX instrument being a little bit slow, and it obviously affects applications like tracking. Can you comment on strategies that could be employed to speed that up?
Yeah.
Perhaps fewer positions of measurements in the initial.
So I don't think it's especially slow. I think most commercial systems have similar time scales. What takes time? What actually very fast is to displace the detection volume, but still somehow there's a small dead time associated with them, which one could try to eliminate because EUDs are really super fast. Or one can use approaches like the group of Stefan Hell did, using interference to shift on instantaneously. So then it takes some time to do the estimation of the position and then to recenter the beam path. And that's the other big time, the big dead time that we found there. So if we were to improve on this, and I think there are technical ways of doing both, we are still limited in the end by the number of photons that we can detect per second, so by the brightness of the fluorophores. So even with very high laser powers, at some point, the fluorophores saturate and we might cause phototoxicity. So this is in the end the limiting factor for tracking. And I think since MINFLUX needs us between two kind of localizations to have a small displacement that we can still follow it, a displacement smaller than the scan pattern size. If you have very fast diffusion, then maybe camera-based tracking is better, because even there, if it jumps by a micrometer, we still not necessarily lose our single fluorophore. And although then we also get, of course, motion artifacts at some point. Yeah, but I think when we are safely below this maximum diffusion, then MINFLUX actually can perform very well. And it's, again, good if you want to have a high precision and long track lengths. Not so much if you want to look at very fast diffusion.
Thank you.
Thank you for the great talk. I have several questions. So first, I depend a lot of membrane receptors on the plasma membrane, and you just mentioned that the control
for the quality of single molecule localization, microscopy is very important, especially I think in the DNA pen case, because we use the transient binding, and actually imaging strand is also like unspecifically binding to, especially I think like lipids on the plasma membrane. So I sometimes feels like I'm shooting in the dark. I saw a lot of like beautifully dots, but I'm not sure if it's like the real signal or it's something artifact. I mean, I still do the kinetic filtering in the post-processing step so that I'm sure that I have proper blinking events of the whole measurement time, but besides that, what control I can do, you would suggest to prefer that my signal is the true signal.
Yeah, which brings us back to the biology of the sample preparation, which is limiting. And indeed, unspecific binding is a problem. And I wasn't aware that DNA probes tend to bind to the membrane unspecifically. I think the first thing I would do is to not have the docking strand, but do the imaging only with the imaging strand to look at the background if this happens and how much it happens. If it happens, maybe one could try a different dye that has a different charge on the imaging strand, which might interact differently with the lipids. Or you try different labeling approaches, not DNA paint, but if you genetically fuse a protein, then there is not much unspecific binding that can happen. But of course, the photon numbers are much less. Yeah, but we struggle with the same, and it's always doing the proper controls, trying different blocking agents, trying different things, even in fixed cells when we do imaging, to reduce the background. Also, what we noticed that it's very important to be super clean with the sample preparation and to really test all the reagents you use, because many reagents, fixative and so on, sometimes have a few fluorophores, which you then see, either as unspecific background or something floating around diffusely. So the negative control without any fluorophore, but entire sample preparation protocol the same is the way to optimize and reduce the background of the dirt or so. Yeah.
Yeah, thank you. The second question related to your tracking data for lifestyle MINFLUX or general from the MINFLUX. I saw that between the movement of the myosin, there are like multiple small direction, like trajectory or fluctuation between the really bigger step they are moving from.
Yeah, okay,
Yeah.
these ones. So
Yeah.
you see them here and you also see them here.
Yes.
If you look at the gray one, this I think it's mostly shot noise, mostly the limited resolution from the number of photons that we detect. So you get this kind of noise. In addition, you see something that looks a bit more systematic like this, or sometimes we see this overshooting here.
Yeah.
Again, we could or should use our simulator to see if this could be an artifact from the MINFLUX itself. I'm not sure about this. Also, we see that when we do careful calibration, that often the noise is not only the shot noise, but if you have a very high precision, that it's movements, still residual sample movements or movements in the beam path that kind of jump on the single nanometer scale, and that's not so easy to overcome. So it could be a combination of both, and that's why we like to look at very bright beats with MINFLUX to establish how stable is the microscope. Do we have any vibrations? Do we have any of these jumps? To make sure that on the spatial scale that we're measuring, it doesn't matter. For instance, here, if you can clearly distinguish these 8 nanometer steps, these half a nanometer movements might not matter so much, depending on your question. Or in the living cell, I think we also here rather limited by the shot noise, or sometimes by nearby fluorophores, by dirt, by something in the sample.
Okay, so you understand like such fluctuation is more like come from the measurement, but not from biology.
Or it could be biology, and that's what many people are trying to do with MINFLUX. And I would always try to exclude that it comes from the measurement before interpreting biology. So, of course, you have these kind of sub-steps, and that's also what the Stefan Hell really beautifully showed in vitro, that kind of the water domain detaches and moves around a bit, and before it attaches again. So you see something like sub-steps. In the end, MINFLUX is not fast enough to really look at fluctuations, conformation changes like this. So it kind of averages out whatever is there, and then it's according to us, to interpret this properly, maybe again with simulations, what we see.
Okay, thank you. Last question, sorry. It's about your endocytosis study. It's really, every time I saw it, I feel it so fascinating. But you detect cluttering and dioning simultaneously in the mammalian cell.
So,
Did you also look at the other label, for example, like AP2, which is related to the last phase of the endocytosis?
So yes, so we do this with our ratiometric dual color imaging in fixed cells using Alexa 647 and then maybe CF660C or another of these far red dyes, where we get a good 3D resolution then in 2 colors. And we spent quite a lot of time also looking at AP2, for example, and together with clustering. In the end, it turned out to be not so easy to get high labeling efficiencies in that case. So now, and this is the future, we take a step back. We are not using antibodies anymore that we use for this project, but we are generating CRISPR cell lines where we have HaloTag, SnapTag to do this labeling more specific and with a high efficiency in 2 colors. So this is at the moment our approach.
Yeah. Thank you.
Ah, also back there. I don't know who was next. Yeah.
Okay, thank you for your nice presentation. And because I'm not a single molecule localization guy, but I still learn a lot from your tutorial presentation. Yeah, thank you. And I have several questions. And my first question is, in the introduction of deep learning part, and I think your training data is simulated, simulated data. And I want to ask, why you use the simulated data and why not measured data? Is there any, difficulty in acquiring the measured data for training, or just you didn't do it?
So it is very difficult because, you get this blinking fluorophores experimentally, but you don't know where the fluorophore is precisely. Of course, we could use a maximum likelihood estimator at low densities to determine where the fluorophores are. But then we still have the localization precision error there, and we cannot easily measure it at high densities. So it's not really any way I can think of, unless one goes to something like these DNA origami structures where one puts very precise fluorophores at well-defined positions. But even then, one has only certain arrangements and cannot show the network all the complexity that can be there. So I think to really have ground truth data is hardly possible in single molecule localization microscopy. And also, if it was possible, it would still be much more work than doing the simulations. And since we can perform such realistic simulations, I think it's also not needed. And the simulations are probably more precise than if we had real data. Of course, there's some limitations. If you have a strongly variant background, if something else is going on, if you train with the wrong parameters, then you get artifacts out of this. And you see them quite precisely by pixels appearing, by a bias of the localization, and then one has to train again.
Okay, thank you. And my next question is, I think in your presentation, and you mentioned about according to the SMLM data, and you designed a model and applied the model to output some. It looks like a blurred molecular structure. And I'm not sure if it is my misunderstanding.
You're talking about the local analysis of super resolution?
I think so, yes.
Okay, let me go to this one. This one.
Yes, yes. And I think this structure is not it's somehow like the prediction structure, right? Yeah, and maybe, okay. And okay, maybe it's just some misunderstanding, I'm sorry. And let's go to the third question. And because I'm a structure illumination guy, and usually in the same, and the result quality is highly depends on the parameter you are selecting, you are using. And I would like to ask about, does the as a single molecular localization also has a similar situation about the parameter selection?
So I think less than a structured illumination, because I think there, if it's wrong, you easily get kind of this honeycomb artifacts or these things. Here, it's important to have a good point-state function model for that localization, because any small difference between model and reality leads to kind of some nonlinear squeezing and deformation in that. If we are not interested in that, then even a Gaussian model performs very well, often even better than an experimental model. And the reason is that an experimental Poisson function sometimes is a bit not straight, but a bit tilted. And then we get the correlation between Z and X. So if the fitter wrongly puts the Z position here instead of here, then it makes automatically an error in X and Y. And there, a symmetric Poisson function like a Gaussian often results in higher precision and higher quality data. So, and this is basically the only parameter. Everything else is standard, or it depends on filtering. But then also it's a bit subjective because we don't get an image out. We get these localizations. And then it's up for us to decide if we want to see all of them or if you only want to see the brightest ones that were localized very well. And that depends on the question. It depends on us if you want to render an image, how we render an image, what spatial scale, to understand best our data, or if we directly look at these point clouds and analyze them. So it's a lot of subjectivity in data interpretation, and many things, many parameters you can set there, but the fitting process is quite straightforward.
Okay, I understand. Thank you very much.
Thank you very much for your talk. You mentioned ratiometric imaging. Could we also use ratiometric imaging with the same dye to detect spectral shifts?
Yes, of course, it would be nice. We tried this once for a long time until we found that the published spectral shifts don't exist, and so we gave it up. But yeah, clearly you can look at it and then look at membrane-binding proteins or other intercalating dyes and see how they shift.
What would be the smallest spectral shift that we would be able to differentiate with ratiometric imaging?
So it depends always on the number of photons you detect. And often these environment-sensitive dyes, they're not the dyes that give us the highest photon counts. But if you detected 1,000 photons, I think some 10 nanometer could be, 10, 50 nanometers could be resolvable. I think it's very easy to estimate this just looking at the Poisson noise and looking at the error one does in the ratios. Also, it depends, yeah, what accuracy you need there in your spectral measurements.
Thank you very much.
Got any more questions? If not, yeah, thanks. Ah, yeah, here, one more.
Okay, thank you very much for the talk. Just a question about the simulator of the MINFLUX. Which program language is based on? If it's MATLAB or Python, maybe? And also another question. Yeah, this is the main question.
So it's both. Actually, we coded it in both languages because the community is still split. Some people like to use MATLAB, the others use Python. And for the biologists who don't have MATLAB, who don't know how to run Jupyter notebooks, we also put it on Google Colab to directly run it then from the internet.
Okay. And I understood correctly that I can also use it as for simulating MINFLUX tracking, not only imaging, right?
Precisely, yes. It's it has you have to define the motion of the molecule and then it simulates it with this motion.
Okay. And just one last question. I would like to know your opinion indeed on the limitation of MINFLUX tracking, especially when it comes to fast diffusion in general, yeah.
In general, so I think it works nicely. You get longer track length, but if it's too fast, as the simulation showed, and as people know in the field, you, on one hand, get very short tracks because you lose the flow force, but even before this happens, you get the bias that you can easily track when it happens to be randomly slower. So you get a bias in the diffusion coefficients and also the bias in the mean square displacement. So depending on what you want to analyze, it's important to be aware of this, especially when you're close to the limit. And yeah, maybe now use the simulator to test what bias you expect. You can expect. Yeah, this is basically it. I think MINFLUX is probably best suited when we want to have a high precision on slower moving particles, because it has the highest precision really when the fluophore is close to the center of the donut, and it can only keep it in the center of the donut when the jumps between individual localizations are much smaller than L. So then it clearly outperforms. I'm not even sure if we are at the limit where the fluophore kind of gets close to the brighter rim of the donut if we are so much more photon efficient compared to camera-based tracking. So it could well be that at some point, and this again one has to probably look at, that one doesn't gain using MINFLUX because one cannot keep the fluorophore close to the center. One doesn't have this great resolution enhancement. It's something to really look at, and if you're in this regime, then maybe camera-based tracking is the way to go.
Maybe just one more. What about the damping effects in the tracking MINFLUX in the MINFLUX sequence? Because when like on the commercial microscope, they suggest to use a certain damping effects, but I don't know if you have an opinion about which is the optimal damping effects with respect to the diffusion you are analyzing.
I think they recommended using a damping, then recommended not using one. What we saw in our simulations, we really looked at this, is that there's hardly any effect on if you use a damping of 0, 1, or 2. So in the end, it seems to not matter much for the diffusion.
Okay, thank you very much.
Good. Yeah, thanks for your attention.